Okay, guys, I'm going to be doing a video on the neuromuscular junction, okay? So, or the synapse between the neuron and the muscle. We're talking about skeletal muscle, okay? So first of all, what I'm going to be talking about is how the muscle cell is stimulated by a motor neuron, okay? So the first thing that I'm going to do is I'm going to draw the structure of the axon, the terminal end of an axon, okay, meeting up with a muscle cell, with a skeletal muscle cell. Okay, so the first thing that I'm going to do is, I'm just going to, this marker here is not the best marker. Okay, so let me get another one. So basically, we have the axon coming down here, and then we're going to have the expanded area of an axon. Okay, that's the expanded area of an axon. Now on this end over here, we're going to have the cell membrane of a skeletal muscle cell. Okay, so what I'm going to do is that I'm going to basically create these folds of the cell membrane. So this end over here, we call that the postsynaptic cell, which is also known as the skeletal muscle. So this is the skeletal muscle cell. Okay, this board is a little shaky here, but it's okay. Okay, so we have here what is known as the skeletal muscle cell on this end. Okay. Now this structure that you see at both ends here and is folded, this is called the sarcolemma. So this is called the sarcolemma. The sarcolemma is the cell membrane of a skeletal muscle cell. Okay. Now notice that a motor neuron, so we're going to call this a motor neuron. So this is a motor neuron. Okay. So now they're going to come together. They're both going to come together, and we're going to call that, and make sure that you're recording here properly. But this is called the neuromuscular junction. Okay, this is what that's called. You know, where this motor neuron is coming together with the skeletal muscle cell, okay? The neuromuscular junction. Or this is called a neuron to muscle synapse, okay? This gap that you see here, this gap is called the synaptic cleft, okay? This is a little gap that we have here between the motor neuron and the skeletal muscle cell. Now, the question is, why do we fold this part right here of the sarcolemma, or the cell membrane of the skeletal muscle cell? And the reason for that is to have more, to increase the surface area so that we can fit more ion channels. We need channels here, okay? Now these channels are going to be called ligand-gated or chemically gated channels, okay? And by the way, this part of the cell membrane, this part of the sarcolemma, or the cell membrane of the skeletal muscle cell, we call that the motor end plate. The motor end plate, okay? That is a specific area on the sarcolemma that has these receptors or these channels that are going to open when a neurotransmitter binds to them, okay? So on this end here, we're going to have vesicles, okay? So we're going to have vesicles in this area, okay? And inside these vesicles, we have a neurotransmitter substance, and that neurotransmitter substance is called acetylcholine. So I'm going to put like little dots in here to represent them, okay? So this is acetylcholine that is being stored in these vesicles, okay? So we call these synaptic vesicles containing a neurotransmitter substance that we need to excite this muscle cell. And we call that acetylcholine, okay? So we're going to write that here. So this is called, this substance that we have in there, this is called acetylcholine. choline, or simply ACH. That's how you abbreviate acetylcholine. Okay. Now, let me show you something else. So up here, we're going to have a channel. Okay, we're going to have a channel up there. Okay, so I'm going to just kind of like make it simple. Okay, and we're going to have a channel here. That channel is for calcium. Okay, this is called a calcium voltage gated channel. That is a calcium voltage gated, or you can say like voltage gated calcium channels. Okay, that's it's better to be to say that. Okay, so this is a voltage. Oops, kind of get ahead of myself a little bit here. Voltage gated calcium channel. Okay, so that channel is actually going to open at a voltage of positive 30 millivolts. Okay, so we're going to have actually an action potential. We're going to have a signal that is going to be traveling along this axon of this motor neuron. Okay, and so I'm going to just draw a line here coming down, and we call this an action potential. So an action potential is traveling, along that axon, and when it comes in this area, then the voltage is going to be positive 30 millivolts, and these channels are going to open for calcium, and calcium will rush into the cell, okay, into the neuron. This is extracellular calcium. So out here is the extracellular compartment, okay? So we're gonna have calcium, coming in here, okay? So this calcium is gonna come in here. And when calcium comes in here, these vesicles are going to fuse, okay? So I'm just going to erase these here, okay? And then draw one of these vesicles here. So we're going to have a vesicle here fusing with this membrane of that neuron, okay? And we're going to have acetylcholine released. So acetylcholine will be released into here, into the synaptic cleft, okay? So this is acetylcholine that is being released. But we need calcium. We need to open those voltage-gated calcium channels to allow for these vesicles to come and fuse with the presynaptic membrane of that motor neuron. And by exocytosis, we're going to have acetylcholine released into the synaptic cleft. Now, on this end here, we're going to have other channels. Okay, so I'm going to just, you can see how many I'm fitting here. I'm going to be fitting a lot of channels here. Okay, so we're going to have channels here, and these are again chemically gated channels. Notice how many of them I'm fitting in here. A lot of them. That's why we folded the membrane, so that we can have many of them. If we was just a straight line, we will have like 1, 2, 3, 4, 5. But here I have like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. So I can fit more of those, okay? So now what's going to happen is that neurotransmitter, that acetylcholine that was released by the motor neuron, is going to bind to two sites here. Okay, to two sites. In each of these channels, you need two acetylcholine molecules to open those channels. Okay, and we're going to have sodium, extracellular sodium, that is going to come into the muscle cell. Okay. So acetylcholine, okay, was released because we were able to open those channels for calcium, and then it diffused across the synaptic cleft, and it bind to these channels, okay, and those channels are going to open, okay. Now these channels are called. These channels, we're going to call them ligand gated channels, okay? And these are channels, ligand-gated or chemically gated channels. They're going to open in response to this chemical, this acetylcholine. So acetylcholine is a ligand. Anything that binds to a receptor, we call that a ligand, okay? So these ligand-gated channels are going to open. Sodium is going to come in. So basically, as acetylcholine binds to these channels, to these receptors here at the motor end plate, the membrane, the sarcolemma of this skeletal muscle cell is going to become more permeable for sodium. Okay, you need to know that for the exam. Okay. So let's read another channel here. Okay, another channel. Now, when the skeletal muscle cell is resting, when it's resting and it's not being stimulated by a motor neuron, then the membrane is going to be polarized. It's going to be polarized, okay? What does that mean? That means that the outside is going to be positively charged, okay? And the inside is going to be negatively charged. So this is what we call the resting membrane potential. For a muscle cell, the resting membrane potential is negative 90 millivolts. Okay, so the resting membrane potential for a skeletal muscle cell is going to be negative 90 millivolts. I know your book, this is a lot of books, they would say like negative 80, negative 85, negative 95, you know, but negative 90 is the number that traditionally is used, okay? So basically, right here, if we record the voltage inside the cell, it's going to be reading negative 90 millivolts. So what happens is that when sodium comes in, the inside is going to turn more positive. Okay, it's going to turn more positive than the outside. So just to go on the other end to represent that, I would just do that. Okay, so here I'm going to put the inside is going to be more positive now, right? And the outside is going to be more negative. Because, you know, we're losing sodium, which is positively charged from the outside. So it's coming in and it's making the inside of the cell more positive. So when we have the reverse of this, okay, then we call that depolarization. The membrane has been depolarized. And by the way, when it is like this here, resting, the membrane is polarized, okay? So the membrane is going to be polarized when the skeletal muscle cell is not being stimulated. But then when it is stimulated, when you have actually sodium coming in here, then it's going to be reversal of these charges pretty much. So the inside is going to be more positive, and we call this depolarization. So the membrane here has been depolarized, okay? Depolarized. Okay. So we went from a state of polarization to a state of depolarization, okay? Now, what's going to happen is that this membrane kind of like, it comes in like that into the interior of the cell, and we call this structure a transverse tubule. A transverse tubule, okay? Or a T-tubule, okay? So that T-tubule is going to take that depolarization signal, which is, you know, actually, it's going to take that depolarization signal into the cell. But before that happens, we need to reach a threshold of negative 55 millivolts. So what happens is that sodium comes in here, and this sodium is going to travel in both directions here, okay? So that motor end plate is going to be depolarized, right? Depolarized, and the signal is going to spread into the cell. But before that happens, we need to open another channel, okay? So right here, we said that the membrane, when it was resting, it was at negative 90. When sodium came in, we're going to bring that voltage into negative 55 millivolts, okay? So we're going to do another channel here, okay? We're going to do another channel here. Okay, and we're gonna let's see, we're gonna do another channel, and so let's do it with this color again. that channel is going to allow more sodium to come into the cell, okay? But that channel is called a voltage gated sodium channel. That channel opens at negative 55 millivolts. Negative 55 millivolts. Okay, so just so you can understand, an action potential was traveling along the axon of that motor neuron. The voltage was driven to positive 30 millivolts. Those voltage-gated calcium channels opened. Calcium came into the neuron. That allowed these vesicles to fuse. Then by exocytosis, acetylcholine is released. Acetylcholine diffuses across the synaptic cleft, and it binds to these receptors. These receptors are known as ligand or chemically gated channels. So ligand or ligand gated channels or chemically gated channels, okay? So we're opening those channels. Who's opening them? The binding of acetylcholine in here. We need two acetylcholine molecules. The sodium comes in, it spreads across the motor end plate here. It depolarizes the motor end plate, okay? And it's going to drive the voltage, which was negative 90 millivolts. It's going to drive it to negative 55 millivolts, okay? And that's going to open these voltage-gated sodium channels, okay? And then sodium comes in here, and then once you open those channels, an action potential develops, okay? An action potential develops. So what we see here is a localized depolarization or a localized potential, and we call that localized potential that is going to drive the resting member potential from negative 90 to negative 55. We call that a graded potential. Okay, so these channels here are responsible for causing a graded potential. A graded potential, okay? Which is localized and expressed in both directions here, sodium. But once we get to negative 55 millivolts, we're going to go from a graded potential to an action potential, okay? So we're going to get from here an action potential. An action potential. It's kind of like that, okay? So now we developed here an action potential. This is kind of similar to neurons, okay? We have what is known as the all or none principle. You either take the membrane to from negative 90 to negative 55, or you don't. In order to do that, you need sodium, you need calcium, you need acetylcholine, you need enough receptors here. Because if these receptors are destroyed in the case of this autoimmune disease known as myasthenia gravis, then we wouldn't have a lot of receptors, and we're going to experience like muscle weakness, okay? But in here, everything's optimal, everything's working fine, okay? So now that we open the voltage-gated sodium channels, we're going to develop an action potential, and it's going to go into the cell. And once it gets into the cell, we get the release of calcium. So the whole purpose of this whole thing that I put in here was to show how a skeletal muscle is stimulated to release calcium. But you can see that there's a number of channels here. We have voltage-gated calcium channels. We have chemically gated channels, which are opened by acetylcholine. That's going to cause a greater potential that is going to drive that or take that resting member potential from negative 90 to negative 55. And then we're going to open those voltage-gated sodium channels, and we're going to have an action potential that is going to take off. It's going to go forward. So this sodium is going to spread this way. So it's going to go forward. It's not going to be like this one. It's going to go this way and that way. It's going to go forward, okay? And then eventually the end result of muscle excitation is to cause the release of calcium, Okay, and then after that, calcium binds to troponin. Troponin is also bound to tropomyosin. Tropomyosin is the molecule that covers the active site on the actin, molecule. And so once calcium binds to troponin, tropomyosin is moved out of the way, the active site is exposed, the head of the myosin filament is, which has previously energized, is going to bind. We're going to cause a cross bridge formation, and then we're going to have pivoting of that head of the myosin filament. It is going to take that thin filament of that actin filament towards the M line, and we're going to call that muscle contraction. Okay, and that causes an increase in tension on the tendons to cause our bones to move. All right, so that's why they say that excitation, contraction, coupling, like it's all happening. In order to have contraction, muscle contraction, you need to excite the muscle. So we mentioned that calcium is stored in the sarcoplasmic reticulum. Okay, but these transverse tubules are wrapping around this sarcoplasmic reticulum, which is by itself, also covering the myofibrils. But at the end, we just want to have calcium being released. Okay, so I'm going to stop the video at this point because I want to show you another video, which is going to talk about how we can graphically, put this together. Okay. All right. So see you in another video.